This week in Biology 1441, we are going to start studying physiology, so the function of organisms. And one of the fundamental things you need to understand to get a grasp of how physiology operates is this important idea of surface area to volume ratios. It's a very important concept that shows up over and over again in many aspects of biology. So in this example, we ask which animal has the higher surface area to volume ratio. So think about it as surface area divided by volume. Surface area meaning the amount of surface on the organism, right, in relation to the volume, the insides of the organism. And you may recall from Bio 1 that surface area to volume is very important for cells, because if a cell has a lot of volume, that means there's a lot of contents inside. It's producing a lot of waste. It needs a lot of energy. It needs a lot of oxygen gas. It needs to get rid of CO2. So it's generating a lot of waste. It needs a lot of food, etc. And the only way things can get in or out of the cell is through the membrane, which, that's kind of like the surface area. How much surface is there? So if a cell has a lot of volume but not a lot of surface, it's going to have a hard time getting things in and out. Think of a city that's really big with only a few roads coming in. It will have a hard time feeding its populace. So we'll have to look at how surface area to volume ratios change as an organism changes size this week and get a grasp on how that works. So we'll start off with a basic experiment with two similar shaped objects, where these will be model organisms. And we're going to fill them up with hot water and put a thermometer in them and see how they lose heat. If they lose heat at the same rate or at a different rate. So we have a small round bulb and a large round bulb, and we'll see what happens. And you can think about what you think will happen. How do you think their surface area to volume ratios compare? Are they the same? Does one have a larger ratio than the other? And will that have anything to do with the experimental results? So we're going to be testing a lot of thermal biology this week. It's a good way to measure how surface area to volume ratios affect objects in the lab, how quickly they can gain or lose heat. Now, in an organism, this is also very important, but surface area to volume also applies to absorption of nutrients and things like that. But heat is something we can easily measure in the lab. So how do organisms such as an ectotherm acquire heat? One way they can do that is through direct radiation from the sun, so absorption from the sun. Another way is conduction, which is the transfer of heat when you're in direct contact with another object, like the rock in this case. How do ectotherms lose heat? Well, it radiates off of their surface, just like it radiates off of the rock, right? It radiates off of their surface into the air. Oops. And that's exactly what happens in the experiment we will do at the beginning of lab. That's radiation as well. But also, they have evaporation of water from their skin or from their nasal passages, their eyes, things like that. That carries off a lot of heat energy. You should probably remember why, if you recall the chapter 3 from Bio 1. Also, convection, the transfer of heat to the air as it blows past the organism, can cool it off. So these are all things that you might be able to test today. So you are going to design an experiment this week based on your knowledge of surface area to volume ratios and how heat transfers to ectotherms, meaning organisms that don't generate their own body heat. So you're going to use models, whether it be clay or those Christmas bulbs or some other object, and you're going to test some aspect of thermal biology. And your experiment may not be directly tied to surface area to volume, but you should keep it in mind while you're designing your experiment. You'll at least have to control for it, if not directly test some variable related to surface area to volume ratios. So you'll have fake fur, you'll have fake feathers, different colored clay, all kinds of things you can use to design your own experiment. You want to keep it as simple as possible, so only one variable, and you're going to want to come up with your null hypothesis and your alternative hypotheses. You'll have thermometers, and after we do that initial experiment with the Christmas bulbs and talk about the concepts, you will be turned loose as a group to design your own experiment. And once you conduct that experiment, gather your data, you are then going to write about this experiment for your second lab report. So in your lab report, remember, in your introduction, you want to use the Google Earth approach. Start zoomed way out. What is this lab over? It's not about a ball of clay with a thermometer in it, right? Or whatever your experiment is. It's about surface area to volume. Why is that an important concept? You need to introduce that thoroughly to the reader. Pretend they are a brand new Bio 2 student and you are teaching them with your lab report. Then you are going to go further and introduce your experiment, why you chose what you did, what your experiment is actually simulating. You're going to tell me your hypothesis and your rationale, which is, once again, reiterating why you chose your hypothesis. Your materials and methods needs to be nice and concise, but tells me what you did. And then your results section with a written result section as well as a figure and caption. And then appropriate statistics as well, either a t-test or an ANOVA analysis, depending on how many tests you do. But you are going to need, I would recommend doing something, a control and a variable, so you'd have a t-test probably, and 3 replications at least. The more the better, but 3 is the minimum. And then your discussion conclusion will wrap everything up, tie in what others have found, some citations. You need to have at least one primary source and your textbook cited. And then you'll have the literature cited section. And pay careful attention to the instructions and the examples online. Make sure you follow the correct format on how to format your lit cited section. All right. So, a few other last things to get you started this week. If you use different colored clays for your experiment, don't squish them together because other labs have to use those clays as well. And once you take blue clay and red clay and squish them together, they don't come apart again. So please don't do that. If you use a piece of fabric for anything or a piece of fur, don't throw it out when you're done. It can likely be reused by other students. Please do not wrap Christmas bulbs in clay. There's no reason for that. Please don't do it. It makes them sticky and gross. And if you use fur, fake fur, and you wrap that around a Christmas bulb, please be sure to take it apart and dry things out at the end of lab so that the next lab has an easy time getting started. Make sure the lab's clean when you leave this week, and we'll have vernier units out on the tables. You can leave them on between labs so the next lab can walk in and get started very easily. All right, so work on those surface area to volume skills. Look it up, maybe Google it. Check out Khan Academy videos on it. You want to be very familiar with this concept. And write a great lab report. Remember, the Writing Center can see you and help you to craft your writing. A lot of students could really use some help on their writing. And then come to us when you need help with the science of your writing. Okay? Have a good week.